Dobrý den. Sorry, that's my knowledge in Russian. I'm Joseph Demeter, representing MERCK, and thank you for the opportunity to be here among the researchers and scientists as a marketing person. And I came with a very optimistic, the title is very optimistic for my topic. Every lab with MERCK CRISPR system. As you know, Sigma is now MERCK. From the agenda, you can see that I will focus mainly on the main products of CRISPR and its features, and I would like also to emphasize how you can reach those surfaces, I mean links, where can be reached all these. Let's start with a background. Here you can see the R&D facility in St. Louis. Here the R&D team, maybe you know Fuqian Chen. Everybody and each company has some background. Let's start with it. In 2002 on Targetron, which is a bacterial system working in some bacteria. In 2007, we came up with finger nucleases with high cost per target. It was the release of the very first targeted genome editing tool. In 2012, we came up with CRISPR-Cas9. We were first commercial supplier of the CRISPR tool, the lowest cost per target, and also with advantage of scalability. And followed 2015, 16, 17 with some other products like epigenetic activator, Zangular lentiviral CRISPR libraries, and synergistic activation mediator libraries. Foundation of CRISPR integration patent for MERCK. In fact, integration of an external DNA sequence into eukaryotic cells using CRISPR grants of patent allowances on all those which are listed here at the moment, and covers gene knock-in technique, which can be used in gene therapy for correction of disease gene, among many other applications. I will skip this because they are general what you already know, was the aim of DSB, of the double-strand break, why we make it. Here the cells own repair mechanism and the tool, bacterial and now the repurposed, which nowadays is used, the 3 components reduced to 2 components. It was formed a loop between the CRISPR RNA and trace RNA, and the 2 components are Cas9 and single guide RNA, as in the literature is used. And let's switch to the genome editing tools, starting with the portfolio. There are different formats to deliver CRISPR. Here you can see the Cas9. Can be different formats for delivery of it: plasmid, mRNA, Cas9 expressing cells, models by virus, protein, and also various formats for guide RNA, like plasmid, virus, where you can do yourself the in vitro transcribed single guide RNA or cRNA, the synthetic guide RNA. And here is the portfolio with, as you can see, the single vector CRISPR regions. I will show it in a couple of slides coming. The dual vectors, CRISPR-Cas9 with the paired guide RNAs, RNA-only formats for embryo microinjection, and lentivirus particles, and Cas9 RNP complexes with cRNA and Cas9 protein. And here you can see their main applications. And of course, we have pre-designed CRISPR and custom CRISPR targets available. And do not forget that we have this link, sigma.com/CRISPRs to find all these. Let's start with one by one. CRISPR all-in-one. Cas9 guide RNA on the same vector. You can see here that efficiently deliver guide RNA, Cas9 and fluorophore in a single plasmid. One vector system with GFP or RFP, that's for the visual confirmation of the transfection, and for modified cell enrichment by cell sorting. And already if you have transfected cell, then you have all the components that are necessary for editing. CRISPR dual vector, these are very flexible systems, expresses Cas9 and guide RNA separately. Independent Cas9 plasmids can be available which co-express GFP or RFP. Here I won't enter in details because you are using it, but just to remember, CRISPR paired cases, Cas9, DNA, and 2 guide RNAs on separate vectors, and due to the functionality or not proper functionality, they were optimized, these paired cases, and guide RNAs were designed in a 5 to 5 prime orientation with PAM spacing between 30 to 150 base pair. And the use of these Cas9 DNA nicking nuclease and paired guide RNAs to increase specificity. And here is the challenge of targeting by CRISPR endonucleases. And the solution was or is expanding the CRISPR DNA sequence recognition tracts to lengths similar to those for ZFNs, gene fingers. You remember that it's 2 times 18, something like 36 base pair target sequence for those. And now this Cas then DNA nicking nuclease in conjunction with paired guide RNAs reduces off-target activity, and also they can virtually target any region in any species. CRISPR injection ready in vitro transcribed RNA can be as mRNA, here, both Cas and Cas9 DNA, and purified guide RNA, which are used mainly for transgenic animal models, and for those who are concerned that the plasmid integrates in the genome. And also Cas9 or Cas9 DNA as mRNA and guide RNA as plasmids. And why this format? Why is its advantage? Because it may result in less off-target effects, can be easier on the cells than DNA, and no random integration of Cas9. And with each the lentivirus, all-in-one vector, permanently integrates genes for guide RNA, for Cas9, and 2 selection markers. As you can see, the operating frame of Cas9 are flanked by Puro and GFP elements. Can be used for difficult to transfect cell types, and also we can see that it can target any sequence in any species. And some features of the dual vector, ideal for pooled and arid screening, great for slow growing and hard to transfect cells. Synthetic guide RNA. or also known the two-part CRISPR-RNA system, which is synthetic CRISPR-RNA and trace RNA. As you know, CRISPR-RNA is one of the components with something like 20 bases, or guide RNA sequence, and tracer, which is a constant sequence. And all the time when the R&D is developing something new, is some rationale behind. Here also it was that to overcome unwanted integration and stress of infection, which is associated with plasmid vectors, that was the reason of developing it. But also we know that the in vitro transcribed single guide RNA solved these integration issues and also improves efficiency. But it takes weeks to produce. I mean, it costs money, time, and that's why it was developed the two-part chemical synthetic CRISPR-RNA, which solve these above problems and can be used with Cas9 protein, Cas9 mRNA, and Cas9 expressing cells. And some of the advantage of this is cloning, promoter, and sequencing free. You don't need to be aware of choosing a promoter. System has immediate activity and less off-target effects. Can target any sequence in any species with this CRISPR-RNA included. Can be chemically modified. It was a feedback from customers that might help or might be important. And you can have it on your desk in 3 to 5 business days. When delivered with Cas9 as RNP complex, cRNA is ideal guide RNA format for modifying cell lines or generating transgenic animals. And we are now at Cas9, which are purified recombinant Cas9 proteins, which in fact shortens the experimental tailends from delivery to expression. And they are, of course, compatible, as I mentioned in the previous slide, with cRNA. Each protein includes nuclear localization signal, which facilitates the imported nucleus, and effective when delivered by microinjection, liquefaction, or perforation. Advantages of Cas9 protein, they are shipped lyophilized. It means can be delivered on ambient temperature. Ready to use, reconstituted protein and form RNP complex seed are ready in 30 minutes to use, and of course they are high quality products. Some variants of the Cas9 proteins are listed here. The enhanced specificity Cas9. Here I'm just showing how were engineered these recombinants. In this case, alanine mutations were made in the chromosome binding motif of Streptococcus pyogenes Cas9. Cas9 detaneinase protein, where it was a RUFC domain modification aspartic acid to alanine mutation, and FN-Cas9 as a CRISPR-based endonuclease. It's like a programmable restriction enzyme, and cleaves target DNA in a staggered pattern, as you know, and it has a 5' overhangs of 4 nucleotide long. And finally, the DCAS9 3' biotin protein for genome engineering and other applications has a 3' epitope and biotin for anti-flag-based or streptovidin-based experiments. And I believe this for researchers and scientists. I put it as an awareness raising, and this proxy CRISPR published data April 2017. I mentioned Fuqian Chen team. It looks that boosted out. It was cited in April 2018. And I made a summary of these. Why we have in fact, a diversity of these proteins listed with different applications, like SP-Cas9 for standard genome editing and cloning applications, ESP-Cas9 for reduced off-target, as also like Cas9 beta A, which is in plus making applications, and FN-Cas9 for cloning, for digestion of DNA, for cloning large fragments and other molecular cloning applications. And I mentioned already the Cas9. Why we include new Cas9 proteins for these applications and also to reduce off-target effects. And advantages of RNP complexes, which was already mentioned, efficiency delivery in case of hard to transfect cells, high specificity, DNA-free, no risk of insertion or mutagenesis, and use for animal applications and transfection. And here I won't comment this new target method, which is called proxy CRISPR. You read it, and for sure it will come, and it will be cited, and new publications will come. And let's see what we can provide you. There are brochures of CRISPR genome editing. You can see here the content. Here it's better. Here CRISPR-Cas9 proteins, its application note, and the CRISPR landing page. You can find it sigmabiology.com CRISPR/CRISPR. And the web content with interesting things like technical bulletin, application form, and it's easy way to reach it, sigma.com/CRISPR, cRNA, CRISPR protein, in fingers. Webinar links, web links, have some additional documents, some trending papers, and papers, press releases, also proxy CRISPR mention. You have this link, and just an example how to order what is included. For example, for cRNA synthetic RNA products for this SP-Cas9 CRISPR research. These are the unmodified products, the cRNA pre-designed CRISPR-RNA. You can have it for human, rat, and mouse targets, 2 nanomole formats, dissolved dry, and shipped in 3 to 5 days. The tracer RNA, which is optimized synthetic tracer RNA, bigger format, HPLC purified, chemical modifications in it, and it's already off shelf. It means you can have it a shorter period of time. But if you want something else, for example, CRISPR-RNA HPLC purified, then you have to go to the custom CRISPR-RNA, and also some different, what is this here? You have to go to custom tracer RNA, but you have to be aware the shipping time varies in this case. And how it looks, for example, a CAS protein product overview, let's call it like that. You have the protein inside, you got in the same box the dilution buffer, the nucleus-free water with glycerol, and the digestion buffer is in it. And this is the list of the protein what we discussed already. And now, how you can buy CRISPR. As I mentioned in the very previous slides, that we have pre-designed guide RNAs. You have to go to sigma.com CRISPRs, and I show you the next slide. Here it is. You just have to follow the instructions. Search. You can search by entering the gene symbol or gene ID, and you can go to reach your product. Or custom designs are available. You just you go on CRISPR@sire.com, and you have to fill this application form, and following the instruction, it's requesting target sequence, project goals, and also they are helping with the application form which are already filled in to be easier to follow it. And here is an example, a real case for gene modification. To create a mouse model possessing a mutation in a gene, is requested to provide gene name, reference sequence. You give the wild type sequence, mutant sequence, and the service is what we are providing in this case. We designed 3 guide RNAs suitable for making the mutation. We measure the cutting efficiency of each guide RNA in novel cell line via cell-1 assay, and we will supply only the highest efficiency guide RNA plus Cas9 in RNA format for embryo microinjection, and also we will synthesize and design the oligodonor required for mutation. And please don't forget that CRISPR is a service, not a product. And as a takeaway message, why MERCK? Years of experience with genome editing. Please don't forget that Sigma is now MERCK. Years of experience with genome editing, technical expertise, assistance behind. The company with complex product portfolio, plasmids, mRNA, lentivirus, and synthetic RNA. First company to introduce the zetafrans, the zinc fingers, and CRISPR available as lentivirus. Thank you for the time. Thank you very much.
Very interesting presentation, and I guess it's very useful. Is any questions, please?
To not shorten the break, maybe you can find me at the MERCK.
I have one short question concerning synthetic cRNA. Especially this is useful web design tool, how you can order your target cRNA. So basically you perform testing on the cell line, and this is cRNA what you supply is for injected in embryo. Are you considering point mutation, especially because, for example, it depends. For example, in mouse embryos, in the same region could be point mutation which will affect efficiency of the cRNA.
I think the team, the scientific team, which is behind of these services, have long time experience, over a decade, and all the instruments and inputs from the literature, I think they are guiding them and following all these.
And what approximate price for one of such targets?
Roughly, but the precise price will be given by our Russian colleagues. But let's say roughly starts from $2,000, and you can have it in 12 weeks.
Thank you very much.
Thank you.
Ну, сейчас